Waardenburg syndrome. Waardenburg syndrome is a group of rare genetic conditions characterized by at least some degree of congenital hearing loss and pigmentation deficiencies, which can include bright blue eyes or one blue eye and one brown eye, a white forelock or patches of light skin. These basic features constitute type 2 of the condition. In type 1, there is also a wider gap between the inner corners of the eyes called telecanthus or dystopia canthorum. In type 3, which is rare, the arms and hands are also malformed, with permanent finger contractures or fused fingers, while in type 4, the person also has Hirschsprung's disease. There also exist at least two types 2E and PCWH that can result in central nervous system CNS symptoms such as developmental delay and muscle tone abnormalities. The syndrome is caused by mutations in any of several genes that affect the division and migration of neural crest cells during embryonic development, though some of the genes involved also affect the neural tube. Neural crest cells are stem cells left over after the closing of the neural tube that go on to form diverse non-CNS cells in different parts of the body, including melanocytes, various bones and cartilage of the face and inner ear, and the peripheral nerves of the intestines. Type 1 is caused by a mutation in the PAX3 gene, while the gene that most often causes type 2, when mutated, is MITF. Type 3 is a more severe presentation of type 1 and is caused by a mutation in the same gene, while type 4 is most often caused by a mutation in SOX10. Mutations in other genes can also cause the different types, and some of these have been given their own lettered subtypes. Most types are autosomal dominant. The estimated prevalence of Waardenburg syndrome is 1 in 42,000. Types 1 and 2 are the most common, comprising approximately half and a third of cases, respectively, while type 4 comprises a fifth and type 3 less than 2% of cases. An estimated 2–5% of congenitally deaf people have Waardenburg syndrome. Descriptions of the syndrome date back to at least the first half of the 20th century. However, it is named after Dutch ophthalmologist and geneticist Petrus Johannes Waardenburg, who described it in 1951. Its subtypes were progressively discovered in the following decades and had genes attributed to them mostly in the 1990s and 2000s. Signs and symptoms. Waardenburg syndrome has multiple different types with some variations in symptoms, and symptoms can vary among those with the same type. The two features consistent across all types of Waardenburg syndrome are some degree of congenital sensorineural hearing loss and some degree of pigmentation deficiencies, most consistently in the eyes. Type 1. Type 1 is characterized by congenital sensorineural hearing loss, pigmentary deficiencies of the hair such as a white lock of hair, poliosis in the front center of the head or premature graying, pigmentary deficiencies of the eyes such as different colored eyes, complete heterochromia iridum, multiple colors in an eye, sectoral heterochromia iridum, or brilliant blue eyes, patches of. Other facial features associated with type 1 can include a high nasal bridge, a flat nose tip, a unibrow synophrys, smaller edges of the nostrils, or a smooth philtrum. Type 2. Type 2. The difference that defines type 2 from type 1 is that patients do not have the wider gap between the inner corners of the eyes, telecanthus/dystopia canthorum. Sensorineural hearing loss tends to be more common and more severe in this type. By far the most common gene to cause this type when mutated is MITF, classified as type 2A. If two individuals with a mutation in this gene have a child carrying both mutations homozygous, for which there is 25% chance additional symptoms are present in the child, such as a hole in the iris, coloboma, small eyes, microphthalmia, hardened bones, osteoporosis, macrocephaly, albinism, and deafness. There have been two known patients identified with mutations in both copies of SNA-ITWO, classified as type 2D. These individuals presented with Waardenburg syndrome type 2, but did not have hair pigmentation deficiencies. When Waardenburg syndrome type 2 is caused by a mutation in so extent classified as type 2E, it can, on some occasions, present with multiple neurological symptoms. These can include developmental delay, early childhood nystagmus, increased muscle tone, white matter anomalies or hypomyelination in the brain, autistic-like behavior, and the underdevelopment or complete absence of many inner ear structures, such as the vestibular system or cochlea. Lack of a sense of smell anosmia due to a missing olfactory bulb in the brain may also be present. Type 3, also known as Klein-Waardenburg syndrome or Waardenburg-Klein syndrome. Type 3 has the same symptoms as type 1 and is caused by mutations in the same gene, but has additional symptoms that affect the arms and hands. These can include joint contractures of the fingers camptodactyly due to underdeveloped muscles, as well as fused digits syndactyly or winged scapulae. Microcephaly and developmental delay are also possible. Type 4, also known as Shaw-Waardenburg syndrome or Waardenburg-Shaw syndrome, type 4 has most of the same features as type 2, i.e. no telecanthus or apparent wider eye gap, but with the addition of Hirschsprung's disease, which is a congenital lack of nerves in the intestines leading to bowel dysfunction. Additionally, hearing loss is not as common as in type 2. Rarely, cleft lip has been reported in this form of Waardenburg syndrome. Type 4 can also be caused by a mutation in so extend the same gene as in type 2E, in which it is known as type 4C. Hearing loss is very common and severe in this type. PCWH, a mutation in so extend, the gene involved in type 2E and type 4C, can sometimes result in the symptoms of both types' neurological symptoms, as sometimes seen in type 2E, and Hirschsprung's disease, as seen in type 4. When this happens, it is called peripheral demyelinating neuropathy, central dysmyelinating leukodystrophy, Waardenburg syndrome, Hirschsprung disease, PCWH. Cause: Waardenburg syndrome is caused by mutations in any of several genes that affect the operation of neural crest cells in embryonic development. Most types of Waardenburg syndrome are caused by autosomal dominant mutations. The few that are autosomal recessive are rare. In most cases, an affected person has inherited it from one parent with one of the dominant forms of the condition. A small percentage of cases result from spontaneous new mutations in the gene, where no family history of the condition exists. The neural crest is a group of temporary migratory cells that are left over after the neural tube has closed neurulation, around the 4th week of embryonic development. They are responsible for differentiating into a diverse group of cells that reach different areas of the body. The neural tube and neural crest are derived from the ectoderm. The neural tube goes on to form the brain and spinal cord, while the neural crest cells eventually go on to form various bones and cartilage of the skull and face by migrating through the pharyngeal arches. They also differentiate into the stria vascularis of the cochlea, the nerves and glia of the intestines, myenteric plexus, Schwann cells, which myelinate the peripheral nervous system to allow sufficient conductivity, odontoblasts, which produce dentin deep in the teeth, some neuroendocrine cells, connective tissue around the salivary, lacrimal. Neural crest cells also have a role in muscle formation, including the wall muscle of certain cardiac arteries. Causes of subtypes. Type 1 is caused by an autosomal dominant mutation in the gene PAX3. PAX3, or paired box 3, is a transcription factor that has a role in maintaining an open window of time for certain neural crest cells, such as those of the head and eyes, to divide and migrate before their terminal differentiation, i.e. to maintain them in the stem cell state. Mutations in this gene therefore prematurely arrest their division and migration, resulting in a minor lack of development of certain face cartilage and bones, as well as underdeveloped inner ear structures and a lack of melanocytes in the iris stroma. Some evidence shows that PAX3 also regulates cells from before the neural crest forms, i.e. the neural tube, since mice with loss of function mutations in one of the copies of PAX3 have neural tube defects such as spina bifida or exencephaly. Type 2 is caused by a mutation in any of a range of genes, the most common being MIT, when it is classed as type 2A. Type 2 is caused by an autosomal dominant mutation in the gene MITF. MITF, or microphthalmia-associated transcription factor, has a more specialized role in the neural crest and is more strictly involved after the neural crest forms PAX3 and so extend have been found to activate MITF. It is known to allow melanocytes, osteoclasts, mast cells, and retinal pigment epithelial cells to divide and migrate. The involvement in osteoclasts explains why mutations in both copies of MITF can lead to bone-hardening osteotrosis, as the osteoclasts are responsible for breaking down bone. MITF also activates transcription of tyrosinase, the enzyme that performs the first step in the creation of melanin-oxidizing tyrosine. A mutation in a copy of MITF can also lead to Tietz syndrome, which is distinguished from Waardenburg syndrome by uniform albinism instead of patchy depigmentation. Type 2B is caused by an autosomal dominant mutation in an unknown gene on chromosome 1 in the locus range of 121-113.3. The gene has been provisionally termed WS2B. Type 2C is caused by an autosomal dominant mutation in an unknown gene on chromosome 8 in the locus of 823. The gene has been provisionally termed WS2C. Type 2D is caused by an autosomal recessive mutation in both copies of the gene SNA2. The study that discovered this association found that SNA2 is activated by MITF as part of neural crest development, and this explained why mutations in MITF cause Waardenburg syndrome, as it results in a lack of activation of SNA2. Mutations in a single copy of SNA2 have also been found to cause patches of hair depigmentation peebeldism without any other symptoms. Type 2E is caused by an autosomal dominant mutation in the gene SOXTEN. Rarely, a mutation in a gene other than those currently known may be responsible for a Waardenburg syndrome with features of type 2. This is usually initially classified as simply type 2, but may be given its own subtype once a gene or locus is identified and established. Type 3 is caused by a mutation in the gene PAX3, the same gene as in type 1. It can be inherited in an autosomal dominant or autosomal recessive manner. It is possible for two parents with Waardenburg syndrome type 1 to have a child carrying both mutated copies of the PAX3 gene, 25% chance, and present with Waardenburg syndrome type 3. A missense mutation has been documented to have this effect. However, it is also possible for Waardenburg syndrome type 3 to present spontaneously with just one mutated copy of PAX3. A deletion of the paired domain region of the gene has been documented to have this effect. However, no major correlation has been found between type of mutation and disease severity. Severity tends to be dictated by mutations in other genes' epistases, as evidenced by distinct familial patterns in severity not tied to Waardenburg mutation type. Mutations in both copies of PAX3 have sometimes led to death before or shortly after birth, and mice with loss of function mutations in both copies of the gene do not survive. Type 4 is caused by a mutation in any of a range of genes, the most common being SOXTEN, when it is classed as type 4C. Type 4 is caused by an autosomal dominant or autosomal recessive mutation in the gene EDNA. Type 4B is caused by an autosomal dominant or autosomal recessive mutation in the gene EDN3. Type 4C is caused by an autosomal dominant or autosomal recessive mutation in the gene SOXTEN, the same gene as in type 2E. A study was done on a rare case of a double heterozygous child with each parent having only single mutations in MITF or PAX3. The effect of double heterozygous mutations in the genes MITF and PAX3 in WSON and WS2 can increase the pigment-affected symptoms. It leads to the conclusion that the double mutation of MITF is associated with the extremity of Waardenburg syndrome and may affect the phenotypes or symptoms of the syndrome. Classification table. Treatment. There is currently no treatment or cure for Waardenburg syndrome. The symptom most likely to be of practical importance is deafness, and this is treated as any other irreversible deafness would be. In marked cases, there may be cosmetic issues. Other abnormalities, neurological, structural, Hirschsprung's disease associated with the syndrome are treated symptomatically. Epidemiology. The prevalence of all types of Waardenburg syndrome is estimated at around 1 in 42,000. Types 1 and 2 are by far the most common, with type 1 appearing to be slightly more common. In a 2015 review looking at 417 patients, type 1 was found to be the most common type, encompassing around half of all cases, 47%, while type 2 was the second most common type, encompassing around a third, 33%. The vast majority, around 85% of type 2 cases, are type 2A. The prevalence of type 2B is unknown, as it was only reported in one 1996 study. Type 2C has so far only been found in one Italian family, and type 2D had only been found in 2 unrelated patients as of 2018. The number of known cases of type 2E that involved neurological abnormalities was reported to be 23 as of 2017, while the number of the rest is unknown. Type 3 is rarer than types 1, 2, and 4, comprising less than 2% of cases. Type 4 appears to encompass around a fifth of cases, 19%. Of its subtypes, type 4C is by far the most common, about 71% of type 4, followed by type 4 19%, and type 4B 10%. It is estimated that Waardenburg syndrome is present in 5% of congenitally deaf people. Congenital deafness comprises around half of deafness as a whole. About 1 in 30 students in schools for the deaf have Waardenburg syndrome. The variable presentation of the syndrome makes it difficult to arrive at precise figures for its prevalence. History. Early descriptions. Descriptions. In 1916, Dutch ophthalmologist Jan van der Hoeb (1878–1952) described a pair of twin girls with deafness and a particular type of blepharophomosis, believed to be the dystopia canthorum found in Waardenburg syndrome types 1 and 3. Blepharophomosis describes eyelids which are underdeveloped such that they permanently cover part of the eyes. In 1926, German physician Irmgard Menn described a family of 4 generations in which 5 children had symptoms of depigmentation of hair, skin, and eyes, deafness, and a mongoloid appearance. Waardenburg later attributed this description to the dystopia canthorum. This later led to the synonym Mend syndrome being recorded in some databases. In 1929, Dutch physician K.T.A. Halbertsma described a familial pattern to dystopia canthorum, and in 1930, Italian physician Vincenzo Gualdi (1891–1976) also confirmed a hereditary pattern to dystopia canthorum. This later led to the synonym van der Hoef-Halbertsma-Waardenburg-Gualdi syndrome being recorded in some databases. In 1947, Swiss ophthalmologist David Klein (1908–1993) first reported a patient with bilateral deafness, pigmentation deficiencies, characteristic facial features, and malformation of the arms. Although this was the first full description of a patient with Waardenburg syndrome type 3, contemporary clinicians did not consider the syndrome he described to be the same as that described by Waardenburg 4 years later, in part due to how severe the arm malformations were in his patient. The syndrome was first fully formalized and described by Dutch ophthalmologist and geneticist Petrus Johannes Waardenburg (1886–1979) in 1951. The condition he described is now categorized as Waardenburg syndrome type 1. Descriptions of subtypes. Type 2 was first established in 1971 when a study noticed that some Waardenburg syndrome patients did not have dystopia canthorum. A 1977 study confirmed a familial pattern to this other presentation. Two 1994 studies first confirmed a link between this type of Waardenburg syndrome and mutations in the MITF gene, now classed as type 2A, located on chromosome 3 at locus 314.1p12.3. Type 2B was first established in 1994 when the same study, which found mutations in MITF in patients with Waardenburg syndrome type 2, also found that some patients did not have any mutations in this region. A second 1994 study found a link to chromosome 1 in the locus 121p13.3. This became known as type 2B of the condition, with the gene designated WS2B. However, it has not been documented since, and the gene responsible remains unknown. Type 2C was established in 2001 when a study of an Italian family with Waardenburg syndrome type 2 features found that they were due to an unknown gene on chromosome 8 at locus 823 that had been broken by a crow. The study established a provisional name for the gene, WS2C. However, mutations in this region in Waardenburg syndrome patients have not been found since. Type 2D was established in 2002 when a study looking to find mutations in the human version of the SNA2 gene known to cause depigmentation in mice found deletions of both copies of this gene in two unrelated individuals with Waardenburg syndrome type 2. Mutations in both copies of this gene have not been found in those with Waardenburg syndrome type 2 since. Type 2E was first established in 1996 when a study identified a girl with symptoms of Waardenburg syndrome type 2, but with additional underdevelopment of the front of the eye, leading to blindness. In 1999, it was found that she had a mutation in her SOXTEN gene, and later studies confirmed the association between mutations in this gene and this phenotype, as well as neurological symptoms such as developmental delay. Type 3 was first given its name by Goodman et al. In 1981 in collaboration with Klein, in which they established the association with arm abnormalities first reported by Klein in 1947. Mutations in PAX3 were first linked to this phenotype in 1992. The comorbidity with Hirschsprung's disease, which would later constitute type 4, was first noticed in various studies in the 1970s. Indian pediatrician Krishnakumar Shah and his associates first outlined the syndrome as a possible variant of Waardenburg syndrome in 1981. The variant was first attributed to a mutation in EDNEB in 1994, now classed as type 4A. Type 4B was established in 1996 when mutations in EDN3 were found to lead to this type of Waardenburg syndrome, and type 4C was first established in 1998 when mutations in SOXTEN were also found to lead to this type. Society and culture. Popular culture. The 2001 novel Shock by Robin Cook mentions a character with the disorder. Enzo MacLeod, protagonist of Peter May's 2006-2017 book series The Enzo Files, has Waardenburg syndrome. His eyes are different colors, and he has a white streak in his hair. In the 2011 Season 6 episode of Bones: The Signs in the Silence, the team must solve a case in which the suspected killer has Waardenburg syndrome. The 2013 book Reconstructing Amelia by Kimberly McCreight features several characters with Waardenburg symptoms. The 2014 book Closer Than You Think by Karen Rose features 3 characters, siblings with Waardenburg syndrome. The 2017 book Murder at the Mayan Temple by M.J. Mandrake features several characters with Waardenburg syndrome. The 2019 novel The Whisper Network by Chandler Baker uses the syndrome as a plot point. Notable people. Canadian YouTube vlogger Steph Sanjati has Waardenburg syndrome type 1. Other animals. Waardenburg syndrome type 2 with a mutation in MITF has been found in dogs, Fleckvieh cattle, minks, mice, and a golden hamster. Degeneration of the cochlea and saccule, as seen in Waardenburg syndrome, has also been found in deaf white cats, Dalmatians, and other dog breeds, white minks, and mice. Domesticated cats with blue eyes and white coats are often completely deaf. Deafness is far more common in white cats than in those with other coat colors. According to the ISPA Complete Guide to Cats, 17 to 20% of white cats with non-blue eyes are deaf, 40% of odd-eyed white cats with one blue eye are deaf, and 65 to 85% of blue-eyed white cats are deaf. Although few studies have been done to link this to genes known to be involved in human Waardenburg syndrome, a genetic disruption to neural crest development would lead to this presentation in cats as well. One of the genes that leads to deafness and a white coat in cats when mutated, KIT, has been found to increase MITF expression. Lethal white syndrome is a syndrome in horses caused by mutations in both copies of EDNA. It leads to death from intestinal pseudoobstruction due to Hirschsprung's disease. A mutation in a single copy of EDNA, however, as in Waardenburg syndrome type 4A, produces the patchy white overrow coat with deafness. Ferrets with Waardenburg syndrome have a small white stripe along the top or back of the head and sometimes down the back of the neck known as a blaze coat pattern, or a solid white head from nose to shoulders known as a panda coat pattern. Affected ferrets often have a very slightly flatter skull and wider set eyes than healthy ferrets. As healthy ferrets have poor hearing, deafness may only be detected by lack of reaction to loud noises. As this is an inherited disorder, affected animals should not be used for breeding. A study of the correlation between coat variations and deafness in European ferrets found all n equals 27 panda, American panda, and blaze ferrets were deaf.